When both transcription and translation are happening, these can happen many times on the same exact piece of DNA or the same exact piece of mRNA. Here's what it looks like when you have transcription happening with many copies of RNA polymerase on the same piece of DNA. And what you can see here is as they're moving along, all these RNA polymerases are causing these mRNAs to elongate, and this is a picture of an actual gene that's going to be very highly transcribed. You can also get the similar thing happening with translation. Polyribosomes, or polysomes, are several mRNAs, are mRNAs with several ribosomes translating at once. Again, for genes that are incredibly important, ones that we need many copies for, this can happen. And so what you can see here is everything's going to assemble as translation's moving along, the polypeptide is going to be growing. This is then going to give basically an empty spot for a new ribosome to come assemble around the start of this particular mRNA in this case. And again, once it moves down this, it's reading this mRNA, this is going to leave a new empty space for a new ribosome complex to come around. And so we do know the structure of many of these different parts. In particular, there's some high resolution studies that have been done on prokaryotic ribosomes. They're just a lot easier to study because it's easy to grow up huge batches of these and do things like find the crystal structures of them. The way that this is done is they basically grow up many copies of this. They'll purify it to a very high purity, and they will make it form a crystal. They're going to then use NMR to bounce off of this, and it's going to leave a note on what structure is at each of these particular locations. And so we know what this entire complex looks like, what the size is, what the shape is at the atomic level. And so doing studies like this really have allowed us to advance and find out many details about how the ribosome functions, the importance of the various different components. So in eukaryotes, though, it is just a little bit more complex. As we saw previously, the ribosome's not that much more complex. It is a little bit bigger, but the ribosome's a little bit larger. It's also a little bit more long-lived than in bacteria. Also, remember, in eukaryotes, there's going to be several other differences. Of course, transcription is going to be separate from translation. In prokaryotes, as soon as something's transcribed, it can be translated. It doesn't need any processing events. There is no differentiation between nucleus and cytoplasm. In eukaryotes, though, remember, transcription is going to occur in the nucleus, and there are several different processing events that need to occur. It needs to have a 5' cap, it needs to have 3' poly A tail, it needs to have splicing happen. And then translation is going to occur separately in the cytoplasm with that mature mRNA that was made during transcription. Also, many of these eukaryotic mRNAs are going to contain a purine, so an A or a G, 3 bases upstream from that AUG initiator codon, which is followed by a G. Okay, we're going to call this the Kozak sequence. And it is similar to the Shine-Dalgarno sequence in prokaryotes. It's just considered to increase the efficiency of translation, interacts with the initiator tRNA, helps everything to assemble appropriately. In general, though, translation is going to require more factors for elongation, initiation, termination than translation in bacteria does. Also, many ribosomes in bacteria are going to be just freely floating throughout the cytoplasm. But remember, eukaryotes, many of these cells are going to have endoplasmic reticulum. And so many of these ribosomes are going to be associated with that in these eukaryotic cells. Now they do have some that are free floating, but they are going to have some that are associated with this ER as well. And there's going to be the rough endoplasmic reticulum.